Hello, and welcome to this weekly overview for Human Physiology Lab. And this is the overview for the Sensory Physiology Lab, week number 4. Again, the objectives for this lab, and these are posted in the handout. And so just starting out with a general overview of the sensory system, looking at sensory physiology. Basically, what we're looking at is kind of a series of structures that are going to be involved with kind of sampling the external environment, sampling, kind of what's going on, kind of on the surface of the body or kind of surrounding the surface of the body. So what we're going to see is that there are going to be a variety of special types of sensory organs or sensory receptors that are found primarily through the skin in terms of general receptors. But then when we start getting into more specific receptors, kind of similar to the stretch receptor we looked at last week with reflexes, we can find sensory receptors deeper within the body, say within, say tendons within the Golgi tendon organs or muscles within the muscle spindles. And so this is just an example of different types of receptors that we can find within the skin that have the ability to kind of recognize a stimulus. And so we have thermal receptors that are able to recognize kind of the changes in temperature in relationship to the body. And again, we have heat receptors as well as cold receptors. We've got Meissner's corpuscles, which are going to be kind of concentrated within the fingertips, giving us a fine sense of touch. Nociceptors are going to be involved with kind of sensing pain, sensing kind of damage to cells and the breakdown products of these cells. And then finally on this slide, we've got the Pacinian corpuscles, which are going to be involved with sensing pressure or vibration within the body. Now, similar to what we've seen where we were looking at the hormonal system and chemical signal molecules that are released throughout the body, in order for the nervous system to be able to respond to some type of stimulus, we need to have receptors being present. And the kind of density of the receptor, the location of these receptors is going to determine kind of the sensory response that we're going to be seeing. And we're going to have an activity with this related to lab. And so if we take a look at a region on the back of the skin, a region of the back, what we're going to see is that the sensory receptors are going to be relatively large, relatively kind of further away from one another. And so because of that, if we do kind of the 2-point discrimination, we've got a point on either end of this caliper. And basically on the back, we could stimulate kind of pretty far away from each other on the surface of the skin and only activate one receptor. And so what we've got is a relatively large receptive field with fewer receptors being present, contributing to kind of a crude sense of touch. In contrast, if we look at a region like the fingertips, we've got kind of much smaller sensory receptive fields and a higher density of these receptors. So because of that, we can have the points of the calipers much closer together, and we're able to have a much finer sense of touch associated with this. And again, we'll be exploring that within lab this week. Now, in the process of converting that external stimulus into a nervous system signal, basically what we're going to look at is kind of a response of these sensory structures, either a sensory cell or maybe a kind of a collection of cells providing a sensory organ. They're going to convert that external stimulus into a nervous system signal that'll be carried along sensory neurons and then carried into the spinal cord. And so these sensory neurons are going to be located with their cell bodies within a dorsal root ganglia, kind of lateral to the spinal cord. And these are going to be cells that are going to extend one process out into the periphery where they're going to interact either at the surface of the skin or interact with a sensory structure. And then another process, which is going to go into the central nervous system and carry that information. So we're going to, stimulate something out here in the periphery, and that information is going to be carried on an action potential all the way into the spinal cord, where it is going to be kind of used to kind of interpret or determine what's going on kind of outside of the body. Now, if we take a look at sensations, we're going to focus in primarily on conscious sensations in lab this week. And so we're going to have a variety of activities associated with somatic senses. The somatic senses, or body, soma for body, are going to be the body sensations. And so, touch, temperature, pain, itch are all examples of body sensations. Proprioception is a specialized body sensation, kind of a sense of where the body is in space. And that's going to be primarily as a result of, Golgi tendon organs and muscle spindles and other kind of receptors that are involved within our muscles, within our joints, within our tendons that are going to give us an indication of where our limbs are in space. And so we'll have a variety of activities associated with that. And then we're also going to have special senses. These special senses, primarily associated with the head, are going to be things like vision, hearing, taste, smell, and balance. And they're going to be, special sensory structures, more elaborate than what we're going to see in most cases out in the periphery. But we're going to have specialized sensory structures, sensory organs involved with the conversion of specific types of stimuli into nervous system signals. Now, in addition to these conscious sensations that are basically going to, kind of come in from the periphery, but then be carried up into the brain region of the nervous system, central nervous system, and ultimately getting up here to the level of the cerebral cortex, where we're going to have kind of conscious understanding of those sensations. So we have kind of interpretation about what's going on in relationship to the body. Now, we're also going to have a variety of subconscious sensations that are going to be present. And subconscious means that they are basically going to be kind of providing information to the nervous system, but it's not reaching to the level of the cerebral hemisphere. So it's not kind of reaching up here to the cerebral cortex. So because of that, it's going to be getting here to the level of the brainstem, or maybe within the level of the spinal cord, maybe eliciting reflexes, but it's basically going to be triggering kind of autonomic responses within the body. And so, regulation of blood pressure, regulation of body temperature, kind of looking at stretch receptors for, kind of regulating blood pressure, regulating proprioception, as well as regulating pH within the body. And again, visual senses as well as proprioception. Now, if we take a look at the four properties that allow sensory systems to distinguish between sensory information, basically what we're focusing in on is this idea that we're going to have a receptor, or kind of a receptor region out here, receptive field out here within the periphery. That information, once it's been kind of interpreted by these receptor cells, is going to be converted into an action potential and then carried into the nervous system. And so what we're going to see is that pathway carrying that information is going to be important for interpretation of that information. And we'll look at some examples of that again within the lab. And so if we're looking at a sensory receptor that is generally associated with kind of touch sensations, they're going to be carrying touch information into the body. And when we activate that receptor, the brain is going to interpret it as if it was receiving touch information. And so the modality is going to be important. The nature of the stimulus is going to be important for activating the receptors and carrying that information along a distinguished path, a determined path within the nervous system. This is going to give rise to the receptive fields, which are often stimulated with a variety of different types of inputs that allow you to localize the stimulus, localize kind of what's going on in relationship to the body, based on the ability of it kind of reaching up to the level of the cerebral cortex and kind of combining that information and interpreting what's going on. Then things like a stimulus intensity and duration will be an indication of kind of the number of receptors that have been activated. So a greater intensity of a stimulus, kind of a greater pressure or greater kind of vibration, greater series of touch, will often increase the number of receptors that have been activated, so activate multiple sensory fields, as well as we are going to see an increase in the frequency of an action potential. And so that increased intensity is going to be associated with more signals coming into the nervous system. The duration of the action potential signals, not the duration of the individual signals, but the duration of the ability of these sensory neurons to kind of carry that information in a series of action potentials is going to be dependent upon the type of receptor that's going to be present. So to make that make sense a little bit more, we can take a look at the properties of either tonic receptors or phasic receptors. And so tonic receptors, kind of illustrated over here on the right-hand portion of this slide, are receptors that are going to be slow adapting. So they'll have an initial firing rate that is going to be relatively high. And so along the bottom of this diagram, we're looking at these little lines, which indicates kind of an action potential that's going to be carried by the nervous system and carry that information into the central nervous system where it's going to be used. With these tonic receptors, what we're going to see is we're going to have kind of a constant series of pressure that's going to be going on for a period of time, the duration of the stimulus. This is going to activate our receptor, and that receptor then is going to trigger the generation of action potentials being transmitted along these sensory neuron processes. Now, in the tonic receptors, we're going to see kind of rapid firing initially, and then the firing will slow down, but it doesn't go away completely. And so think about it. If you, are kind of changing a tire and the jack comes out from under the car, the tire rests on your hand, you're going to have some pain stimuli that's going on, kind of related to the tissue damage that's occurring because your hand is essentially underneath the tire, kind of supporting the weight of the car. You don't want to forget that your hand is there. You want to remember that this kind of damage is occurring. You want to remember that you've got this painful stimuli and be able to respond to it. And so we want to have tonic receptors for areas of like pain. Other times, we're going to have phasic receptors, and phasic receptors, again, in response to a constant stimulus, will be kind of activating our receptor, but they're only going to show kind of a series of action potentials being generated early on. And if the stimulus remains constant, we're no longer going to see additional firing of these action potentials, kind of additional kind of signaling of the stimulus that's going to be occurring. And so an example of that would be like the clothes on your body, or putting the ring on a finger on your hand. You feel the pressure, you feel the presence of the ring or the clothes on your body initially, but very quickly, unless you kind of move the body or spin the ring or kind of move your clothes, which case you're not looking at a constant stimulus, you're changing the stimulus, you're going to see that the receptors are basically going to stop sending that information because you're not going to want to overwhelm the nervous system with information that's not vital to what's going on. Now, in terms of sensations, again, focusing initially on these somatic sensations, they're coming from the body. So coming from the skin, coming from the viscera, the kind of internal organs, as well as coming from muscles. And we looked at examples of muscles last week when we were looking at reflexes. So the four somatic sensory modalities will be tactile sensations, which is a scientific way of saying touch; proprioception, which is going to be involved with kind of the body position in space; temperature, again, related to kind of hot receptors or heat receptors or cold receptors; and then finally nociception, which is pain reception. That information is going to be coming in from the periphery, kind of activating these receptors carried by the processes of these sensory neurons sitting here within the dorsal root ganglion, and then transmitting that information into the spinal cord. Now, in some cases, that information is going to trigger kind of localized reflexes at the level of the spinal cord, similar to what we saw last week with our myotactic reflex. Other times, that information is going to be carried up ascending within the nervous system, where it ultimately will be kind of processed by cells within the brainstem, or in the terms of conscious sensations, it is going to be carried by cells within their cell bodies within the thalamus, carry that information up into the cerebral hemispheres, the cerebral cortex, where we're going to have conscious perception of the sensation going on. In terms of the special sensations, again, we're going to have things like the eye, where we have specialized sensory receptors along the receptors along the retina at the back of the eye. We're going to have smell and taste, where we're going to have specialized receptors either within the nasal cavity or taste receptors kind of along the surface of the tongue. And they're going to be able to respond to, kind of chemical shapes associated with our foods. And then finally, we've got the ears here with the semicircular canals, which are going to be important for vestibular sensations, kind of the perception of movement and balance. And again, we'll have activities associated with this in the lab. So this week in Physiology Lab, what is it that we're actually going to be doing? So once again, we're asking you to download the Google document, save it with the naming convention that we've been using throughout the course, and it's available as a Google document. So you can download it to your computer to edit it within Microsoft Word, or you can save a copy of it to Google Docs and be able to fill out the report using the Google program available. And so that is going to be available there. As always, we've got a PDF version of this lab report and lab instructions in case the version you've got gets edited and you need to refer back to that. Now, if we take a look at the specific activities in this week's lab, the first activity is going to look at gaze convergent. And so this idea that we are, individuals with binocular vision. We have two eyes. The eyes are not, kind of attached to one another, not overlapping. They're, kind of separated by the nose, kind of on either side of our face, either side of our head. And so if we look at an object that is relatively far away, we're looking at the fact that both of these eyes are going to be looking kind of at a distance. But as the object begins to become nearer, our eyes have to kind of converge. Our eyes have to kind of angle to the inside. And so, kind of aim towards the nasal region, aim towards the nose. And so your left eye is going to be aiming, kind of towards the right. The right eye is aiming kind of towards the left, kind of looking, as if towards that nose region, the nose being kind of at this point right here, which allows them to kind of focus in on a near object. And so what you're going to do in the gaze convergence is kind of alternate looking at objects kind of near and far with both eyes and kind of observe what happens. In terms of accommodation, again, this is kind of related to what we had with gaze convergent in the second activity. We're looking at the ability of the eye to respond and keep an object in focus as it's, coming nearer to you or potentially moving further away from you. The third activity is looking at the blind spot. And so again, looking at vision. So at the back of your eye, we're going to have the sensory structures within the retina. So the rods and cones that you would have heard about in other courses. But those rods and cones are going to be, converting that light information into nervous system signals, into action potentials. That information is going to be carried by, specially sensory neurons, and those neurons are going to have processes. They're going to exit the retina at the area of the optic disc at the back of the eye. And the region of the optic disc is not going to have any of these light detecting photoreceptors. So in essence, you're going to have a blind spot. You can have a region of your eye where you are not going to have these receptors that are able to respond to the light signals. And so that kind of contributes to the blind spot. Activity 4 is looking at sensory modality. And so generally, our photoreceptors at the back of the eye are going to be responding to, kind of visual stimuli. They're going to be responding to kind of light images and light patterns by, again, converting that light information into nervous system information. However, we can kind of mechanically stimulate the retina by closing the eye. And again, don't put too much pressure on it. But with the eyes closed, rubbing them slightly, you're going to be able to see kind of little, like little fireworks going, little kind of sparks of light that are occurring. Again, because you are kind of non-specifically activating the photoreceptors. And the way that the nervous system is organized is that the visual information carried by the optic neuron is, these nerves are used to carrying visual information. The nerves that they synapse on are used to receiving visual information. And so when they're activated, when these photoreceptors within the retina are activated using a different modality, in this case, pressure, the nervous system is going to interpret it as if it is visual information. The fifth activity, again, looking at photoreceptors, is the idea that over a period of time, photoreceptors are going to begin to lose their sensitivity. And in doing so, we're basically going to have the ability to generate a negative after image. Basically, this idea that as you stare at an object, as your photoreceptors lose their sensitivity, if you move on to kind of a blank object, a white piece of paper, what you're going to see is kind of the inverse of the signal that's being present. And so on this example, if you focus in on the dot, you've got the little pink dots that are kind of moving around. When the pink dot goes away, what you're going to see is a green after image. Now, if you pull back and look at this object, without focusing on the dot, you can see, that the green dot isn't necessarily there if you are not overstimulating the photoreceptors and causing them to lose their sensitivity. And there's some additional resources on the Canvas site, some images that you can use for these negative after image kind of activities. The sixth activity is the 2-point discrimination. So, don't do it on your tongue, but we're going to have calipers, which will allow you to kind of adjust kind of the distance between the 2 points. If you don't have a caliper available, you can kind of bend a paper clip and then just have it at different distances and measure kind of the density of receptive fields, basically the size of the size of the receptive field and the kind of location of the receptive fields. And what you're going to focus in on is how close apart can these points be to one another and still be recognized as 2 distinct points. And what you expect to see is that we'll have much finer sensation, much smaller receptive fields in areas like our fingertips, as opposed to, areas kind of on the, on our back or, potentially on our legs or a region like that, back of our arms, perhaps. Activity number 7 is looking at thermal illusions. And so this is looking at temperature sensation and the process of sensory adaptation. So the idea is that you're going to have 3 containers of water. You have one which is very cold, one that is very warm, and then a series of water that's at room temperature. And so, we're going to look at what happens when you take your hands that were in cold water and put them into room temperature, a hand that's in warm water, and put it into the exact same temperature, and seeing how your body is able to interpret that. There are some additional resources for that, some additional videos showing this thermal illusion available on YouTube. Activity number 8 is looking at the relationship between taste and smell in our interpretation of flavor information. So, why is it that if you are congested or if you have a head cold, foods don't taste exactly right? And so we'll take a look at that. Again, some taste activities within the lab, as well as some video resources if you are interested in looking at this a little further. And again, these additional resources aren't required, but they're available to you if you want additional information. Activity number 9 is taking a look at our semicircular canals. These are going to be important for our vestibular system involved with maintaining balance and sense of movement. And so generally this would be tested in kind of a clinical setting within a Barony chair, but we can do the same type of things with a swivel chair. So, you can put your subject into a swivel chair, start to spin them around. Please don't get wild with this. We don't want people kind of flying off the chairs and hitting their heads on things. So if you are spinning them, perhaps have two students involved with the spinning, kind of move on either side of the subject that you're spinning so that they start to lose their balance as their vestibular system gets activated. You can, catch them before they fall and potentially hurt themselves. Once again, other examples of activation of the semicircular canals, true Barony chair activities available on YouTube with links that are posted on Canvas. And then the final activity was taking a look at nystagmus, and we got an example of nystagmus in the GIF over here on the right-hand side of the slide. But these are repetitive, uncontrolled eye movements, kind of oscillating back and forth like this. And we're going to see, how we're able to stimulate that primarily through activation of our vestibular system. And how the body is responding to trying to keep the gaze kind of focused on a specific location, even when the body is in motion. And if you have difficulties eliciting this within your test subject within the laboratory, again, we have some additional videos that are available on the Canvas site. So the goal of this is that you'll go through these different activities within the lab, fill out some answers, your observations as you're going through, fill out the questions at the end of the lab report, and then submit it onto Canvas again at the location submit lab report number 4 here, and that'll be available for your lab instructor to kind of give you feedback on. As always, your lab instructor may have additional information available to you. And so please see if they have any changes or updates for the lab for this week. As always, if you have any questions, please do not hesitate to reach out to me. Thank you and have a great week in lab day.